you. Let's see. Put that on 4. Well, it was a lot of fun to think about following Mara's talk, actually, for a couple of reasons. The first reason is that at Yale, one of the sort of pet projects I have is that we're building this really advanced new biology building. And a bunch of my colleagues carved out part of it to create this quantitative biology institute. And, I'm sitting there thinking, well, my work's pretty quantitative. And I loved how Mara's work showed that, by applying math to really just anything that you could observe in detail is going to mean that all biology is increasingly becoming quantitative or Q-bio. So it just sort of made me laugh, thinking that we're going to have to expand it throughout our whole building. The other thing is I've never been in the same session with somebody who's also had 4 kids. So both of them, between the two of us, there's a lot of children that have accompanied everything that you will see in this afternoon session. So I think that's kind of amusing, actually, if not, when I really think about it, seemingly impossible. Let me just get this guy working. There we go. Okay, so this is completely different. So, but again, very much an example of how when you follow the details of a process that you are very interested in, you can go from the sort of arcane sort of aspects of, in my case, a sort of chemical biology phenomenon, all the way to something that is soon going to be in the clinic as well. And in my case, I've studied how RNA recognizes other things. Many of you may know more about our work in which we've solved the structures of very large RNA molecules and tried to figure out the sort of alphabet of molecular contacts that can glue RNAs together. But this is going to be a talk about how proteins recognize RNAs, and in this particular case, about how our innate immune receptors recognize RNA molecules, and then how we can sort of harness our knowledge of that to create new agonists for jump-starting our innate immune system. So today we're going to be talking about one of my favorite molecules, which is retinoic acid-induced gene 1, or RIG-I, which is sort of your first line of defense against a very broad group of extremely nasty viruses. You definitely need this. If you don't, it's lethal. If you don't have it, that is. But the funny thing about RIG-I and its cousins is that they're not just your regular RNA binding proteins that just stick to RNA. They're what I increasingly think of as smart RNA binding proteins that can also bind ATP, which causes them to wiggle around, and also couple that RNA binding to other processes. And so, as it happens, RIG-I is a member of what used to be called helicase superfamily 2. Although you'll soon see, we increasingly call them RNA-dependent ATPases because most of them are not unwinding enzymes or helicases, and this is one of those examples. Most members of the so-called superfamily 2, as we'll call RNA-dependent ATPase superfamily 2, contain a number of conserved motifs that have a very similar architecture. Each one of these regions of a superfamily 2 protein has a structure that's superimposable with the protein REC-A, which, as you guys know, is involved in recombination and repair. And these REC-A-like folds come together. They make a little cleft that binds ATP, and RNA lies along the top. And those things associate through very conserved sequence motifs within the protein. If you want to know about a true helicase that really rips things apart, you're not going to hear about it today. In our case, today we're going to be hearing about an ATPase that instead uses the power of RNA binding and ATP hydrolysis to change conformations and influence signaling. But if you really love helicases, you can read this paper where we have identified a particularly great helicase that's in a particularly fulminant strain of hepatitis C virus. And we've linked how good of a helicase it is and how well it strips RNA apart to how well that virus replicates. So that's another story if you're really into that. And the way most RNA-dependent ATPases in this family work is shown in this silly cartoon movie made by my good friend Suum Yang. When we did single molecule analysis on the hep C helicase, we saw that two of the domains that hydrolyze ATP creep along one of the strands. It's really a single strand translocase, and they drag this other domain behind it, which kind of rips things off the track of the single-stranded RNA. And that could either be a second strand of RNA, or it can be a protein, it can be anything. These are really molecular wire strippers in this family. And using crystallography, single molecule studies, and many other things, we've watched the scissoring motion of these domains and how it creeps along RNA and accomplishes these mechanical tasks. So I was sort of working away at that family for a long time. And then I got a great postdoc, actually, who came from Germany. He was from Dresden. His name's Eckhard Jankowski. He's a sort of famous guy at Case Western now. And Eckhard took all the sequences of the superfamily 2 RNA-dependent ATPases, and he made a nice tree, an unrooted tree of them. And he noticed that all the ones that we'd been studying that were nice unwinding enzymes all grouped together in this one branch. But there were other members of the family that had similar sequences, and it was becoming clear that they might not actually have any propensity to unwind DNA or RNA, or even move on it. And a particularly interesting branch that was a little farther away, he classified, which differently at that time, but is now classified as the RIG-I-like proteins. And this is a pretty important group of proteins. We now know that it includes the innate immune receptors RIG-I, MDA5, and the helicase domain of dicer, metazoan dicers. These are all proteins that bind double-stranded RNA and utilize ATP in some way to accomplish some sort of complex task. But anyway, so we knew that there was a group of these proteins that segregated away and that had some profound differences in their overall organization and in their sequence motifs that were usually conserved. And this became really clear when I was, Eckhard and I were at Columbia University at that time, and one of our colleagues named Paul Fisher, who was working about 5 floors above us, took the elevator down and announced that he'd found a helicase that was involved in apoptosis. And we're like, that sounds kind of interesting. And he asked if we could figure out if it indeed unwound RNA and hydrolyzed ATP. He'd found it, interestingly, in a screen for genes that suppress melanoma growth. And he called it MDA5. And then we published this in 2002, and we saw the weirdest things with this protein. We found that it did not convert ATP to phosphate if you gave it single-stranded RNA, which is what you would normally see as the substrate for a superfamily 2 helicase. Instead, it only hydrolyzed ATP with double-stranded RNA. And we saw that it could not unwind RNA. It was acting totally differently. It was acting more like a G protein. And the really weird thing about this protein was that it had these caspase recruitment domains stapled onto it. And we thought, well, what's a death domain doing on an RNA-dependent enzyme? Somehow it was linking RNA metabolism to cell death and regulation. So this was pretty interesting, and we kind of, it's funny, because in that old paper we hypothesized, we said this would make a really good viral RNA sensor. And then a year later somebody showed that's what it actually does. So then cousins of MDA5 were identified, including the protein called RIG-I, and they were shown to play a key role in sensing double-stranded RNAs and viral RNAs in the cell. And as the rest is history, dicer-like helicases are really important in small RNA metabolism. So here we had this protein family that was really important. It had ATPase activity that was stimulated by double-stranded RNA, which made no sense, really. It had other weird enzyme behaviors, but there was no structural information on any member of this family. And so the field sort of hit the wall. People speculated a lot about how these proteins worked, but it became increasingly Baroque levels of speculation in the absence of any structure. This is in general true for innate immune receptors. Just to backtrack a little bit so that you get an idea about what pattern recognition receptors and innate immune receptors do, is that in your cytoplasm, you express a huge menagerie of different proteins whose sole job is to sense molecular determinants of pathogens that get into your cells. So these could be weird carbohydrates left behind by pathogens and microbes that cause infection. They can be they can be different kinds of lectins. They can be DNAs, RNAs, and we have a special sensor for each of those sort of molecular calling cards that are left behind by pathogens. So there's a huge family of these. There's very little structural information on any of them except for two groups, the STING and RIG-I-like receptor pathways. But at the time we started this, there was no information on this group, and this is the major group for, as I said before, detecting RNAs that are the result of viral infection. So we came to focus on RIG-I because it's actually the most important of the receptors in this family. It's an essential component of your immune system. If it's dysregulated, it becomes hyperactivated, as does MDA5. When that happens, you get autoimmunity. It has emerging roles in development and differentiation. And if triggered, it can elicit potent anti-tumor responses that are of great interest right now. But this is their normal target, and we definitely need them against these things. And if we could really understand the molecular basis by which it worked, we wondered whether or not we could harness it to create specific vaccine adjuvants, antivirals, and cancer vaccines. So what is the pathway? When a virus infects, it dumps double-stranded RNAs, in some cases, sometimes single-stranded RNAs, into the cytoplasm. If that double-stranded RNA has a triphosphate that's still accessible, RIG-I, which normally is floating around in an auto-repressed state that I'll show you in a minute, binds this triphosphorylated double-stranded terminus, and when it does that, it flips out these death domains, which interact with a receptor called, an adapter protein called MAVS, and through a pathway that we are not going to discuss because we don't have time, it leads to a very complex signaling cascade and turns on the production of type 1 interferons and a bunch of inflammatory cytokines. Importantly for later, nobody has ever been able to just trigger the action of one innate immune receptor at a time. Most of the ligands that people use to turn on these processes turn on both RIG-I and MDA5 and all the toll-like receptors, everything all at once. So we were curious about first of all, if we could figure out how RIG-I recognized RNA, and then if we could use that information to only turn on RIG-I, so we could understand just that one guy, as opposed to this big menagerie. What was known when we started was that it did seem to prefer double-stranded RNA. Most people believed it only signaled when you gave it very long double-stranded RNAs, and that held the field back for quite a while, because that was the context in which people tried to crystallize them. And there was good evidence already that it needed a 5' triphosphate. And it had to ignore single-stranded RNAs and RNAs with O-methyl groups and a cap structure, and as we'll see, monophosphate RNAs, which are really abundant in our cells. Anyway, the field was kind of messy, and then we got to work trying to crystallize RIG-I. And we did this by kind of ignoring all of the literature, and we set out to overexpress RIG-I, human RIG-I, and we found a construct that was nice, soluble, and well-behaved. And we then did our own biochemistry on which RNA ligands had the best affinity. And as it turned out, it really liked little double-stranded RNAs that were about 10 base pairs long, not big guys. And with that knowledge, it crystallized like a shot. And we got this first crystal structure of a member of the family. And you can see in green, RIG-I wrapped around a 10 base pair RNA, wrapping around like a donut. You can see the one end of the RNA sticking out the back. And here it's sticking out the back, and you can see it threading through the middle. And one of the things that was clear from the structure was that there's no perturbation to the A-form geometry of the RNA. This was at very high resolution. On the other hand, we only got one structure with this one RNA. When this was deposited in the PDB and we were getting ready to publish, we had two colleagues that nearly had a heart attack because they also had structures of RIG-I, and they were Marco Trigiano and Stephen Cusack. And we all figured out at a meeting one summer that we had structures of this thing. And so we got together at the meeting, and it was like one of those westerns where you have to show your cards, those movies. And so all of us like showed our structures, and it was fantastic because they were all so similar. And then we were able to sit down and name the different domains together so we have a common nomenclature for the architecture of the protein. And as it turned out, my colleagues had captured this in some slightly different states with a few differences in the ligands. So within the span of a few months, all of a sudden there was a big group of new structures that helped us understand the architecture of this family. Anyway, we all published within a week of each other. Me and Stephen published together in Cell, back to back. And what we can show you is that RIG-I has a fascinating architecture. Here's the RNA duplex in pink in the center. And in the interest of time, we're going to have to whip through this pretty quickly. The so-called helicase or ATPase domains are shown here. That's the only conserved part. The protein and all members of this family have some new structural innovations that are really important. One of them is this HEL2I domain, and it's like an alpha helical bundle that just sort of sprouted out of one of the helicase domains. And that innovation enables members of the family to bind double-stranded RNA. You can see this comes around and binds both strands of the duplex. One other important innovation is the C-terminal domain, which is this little zinc binding domain. And as we'll see in a moment, this is essential for recognizing the 5' triphosphate. And it comes around and caps the terminus of the duplex. And my favorite domain is what we called the pincer domain, which everyone prior to this called a linker, and everyone assumed it was unimportant. This unimportant linker turns out to be this, what we call a molecular camshaft, and it transduces the information in the binding of the CTD into the ATPase domain, and it's how you coordinate the information about ATP binding and hydrolysis into together with the binding of the viral ligand, and that will have implications for signaling. So that was a happy thing, but what we didn't visualize in those first structures is that the card domains have to be present, and they're actually here. They're not in my construct. And these guys have to interact with MAVs to trigger that cascade. So this is still an active area of research. I'll just show you some models for how we think this works. We know that if you just have the RIG-I core, the ATP and RNA binding core, and you don't have the C-terminal domain, the DEF domains will park on the outside of the alpha helical bundle, this HEL2I domain. But we know that when RNA binds, they get ejected because their position is incompatible with the interactions of the CTD with the RNA and with this domain. So when RIG-I wraps around its targets, and especially once it binds ATP, we believe that's the trigger for ejecting these cards. And right now, that's a process that is best visualized with things like FRET and other dynamical techniques, and those are going on in the lab right now. So RNA and ATP initiate signaling. So the first part that I'll tell you about is RIG-I and ligand recognition. We set out to understand how RIG-I specifically recognizes viral RNA and to develop an optimal activator, and at the end, hopefully we'll have time to show you why RIG-I is not turned on by monophosphate RNAs. And then hopefully we'll talk a little bit about the implications for clinical work. So in the very first structures, we saw that the double-stranded RNA target was not perturbed in any way. And there were lots of contacts to moieties that are only present in the RNA. Specifically, 2' hydroxyl groups are one of the most important interactions from highly conserved amino acids in the protein. And it's interesting because there are really not too many electrostatic contacts with the phosphate backbone. And that's important because this needs to be an RNA sensor. It can't start picking up on DNA molecules as well. It's very sensitive. And you can see the interactions with the CTD are quite important. The CTD parks on top of and stacks on a terminal base pair. And that's why RIG-I is specific for blunt duplexes like you have at the end of a flu virus panhandle. You see a network of interactions here. The footprint is 10 base pairs. And this is just to quickly show you that this HEL2I domain, that innovation that helps you with double-stranded sensing, if you capture it with different nucleotide binding states, you can see it sampling along that 10 base pair span. And we already talked about the pincer domain and how it sort of folds around the ATPase domain to couple interactions up here with ATP hydrolysis. She's going to try to find the phone in somebody else's coat, which is always a challenge. So at this point, it was really still unclear how Rig-I recognized triphosphorylated RNAs. So then my lab got to work on trying to crystallize it with something that was much more of a mimic of a virus panhandle. So a hairpin RNA with a triphosphate. And we succeeded, and we solved the first structure of sort of an intact Rig-I ligand. And you can see that the CTD forms a network of really nice contacts with the alpha and beta phosphate. Interestingly, not with the gamma. And this was interesting because it suggested to us that the primary ligand for Rig-I was actually not a triphosphate, but a diphosphate. And as I'll show you later, diphosphorylated duplexes are actually even better ligands and agonists for Rig-I than triphosphates. But we were beaten to the punch by Günther Hartmann's lab, who saw this result, made the diphosphorylated RNAs, and actually showed in vivo that they're wonderful agonists. And they got a paper in Nature for that. So I was happy for them, but sad we were slow. But anyway, so we were able to visualize these interactions, but we wanted to show whether or not they had energetic content. And so we do a lot of quantitative biochemistry in my lab. So one of the things we do is a lot of sort of thermodynamic studies and direct binding studies. So this is showing you how you can watch a radio-labeled RNA form a complex, a one-to-one complex with Rig-I. And you can plot this data. This is a gel shift experiment. And you can see, if you look at these concentrations, this is a very high affinity interaction. And it shows that if you have a triphosphorylated double-stranded RNA, you get picomolar binding affinity, whereas if there's no triphosphate, it's in the mid-nanomolar. And you can actually initiate signaling with something like this if you dump enough of it in. So this study was important because it showed that it's really all about affinity primarily, although I'll show you there is some differences in how efficiently you can initiate the signaling conformational change. But this guy has evolved the ability to sense just tiny amounts of potentially viral RNA in the cell. And you have to have that in order to be sensitive for the target ligand. Okay, so at the time, people were still saying, oh, well, Rig-I forms filaments on giant RNAs, and that's how it signals. So we did experiments like I showed you on the last slide with RNAs that varied in length from 10 to 500 base pairs, and we also looked at single-stranded RNA, because there were still papers appearing saying that Rig-I could signal on single-stranded RNAs with triphosphates, and we did all of this work. One innovation that was important was that RNAs like this could bind Rig-I at either terminus. We wanted to create a new family of RNAs for study of Rig-I that would confine Rig-I to only one of the termini. And for that, we started to create these hairpin or stem loop RNAs, which can't bind down here but bind here. And we varied their length from small to large. We did the binding studies with the radio-labeled ligands. And just as in the slide I showed you before, you don't get any increase in affinity when you expand the length of an RNA duplex any greater than 10 base pairs. So there was no advantage to larger RNAs, which came as a big surprise. And I can show you, too, that we didn't just look at direct binding. We looked at the ATPase activity of this enzyme as a function of RNA to sort of get a Km for RNA or a functional RNA affinity. And you can look at the observed rate constant for ATPase activity as a function of RNA concentration. And you can see for a long double-stranded RNA that has no triphosphate, you have kind of low affinity, and there are issues with the overall behavior. And if you have no triphosphate, you get some affinity, but you get the most affinity and the most activation of the enzymatic activity of Rig-I with a triphosphorylated duplex that's at least or greater than 10 base pair. So this suggested to us that the activating camp for RIG-I and ATPase activity was a blunt 10 base pair duplex RNA that had a triphosphate. And we saw, again, much to everyone's concern, that it bound as a monomer, not as a multimer on these RNAs. But the real question is, would these actually cause signaling? We had these cool little hairpin RNAs, so the question was, were they biologically active? And to test that, we used a cell-based assay that was designed by others that's kind of clever, in which you take 293 cells, and you basically transfect these with plasmids that contain luciferase reporter that has a beta interferon promoter in front of it. And this 293 T cells do not express a lot of endogenous RIG-I, and that's useful because then you can have a plasmid encoding the receptor. You can also put that in. You cook that for a bit. If you don't add RNA, you see no signaling. The cells just grow as normal. But if you challenge with an RNA that is going to activate signaling and activate the interferon induction response, you're going to see luminescence. And what you see is something like this. So for example, if you add no RNA, you don't get any relative luciferase units. But with these little hairpins, we saw very robust interferon activation. And we saw this in a variety of cell types. And so you can compare all different kinds of RNAs in cells in this type of assay. You can use other kinds of cells and use qPCR to monitor interferons or cytokines, which we often do, A549s and other kinds of things. But you can see here that if you have an RNA that's the same length as this, but which does not form any base pairs, this nsRNA that has a triphosphate, you get no interferon induction. If you have a double-stranded RNA that only has a hydroxyl, you get very little, if any, induction. But if you have a 10 or 14 base pair duplex, which is the ones we use the most commonly, with a triphosphate, you see a monster amount of interferon induction, much greater than had been seen with just standard double-stranded RNAs or with poly-IC. So we were very excited, and this suggested that indeed RIG-I can function by binding something that looks very much like the terminus of a flu panhandle and activate the entire cascade by just forming a simple one-to-one interaction. And that we had, we don't have time, but we showed in other studies that this process does not require ATP hydrolysis, which was another kind of surprising thing because people hypothesized that because RIG-I looks sort of like a helicase, it would need to move on the RNA to cause this response. We haven't seen that, but it does need to bind ATP to give the optimal response. So we think it's all about forming an active complex. So that was all very nice. In a very contrived cell-based assay, we had shown we could turn on a reporter for interferon. So you could say, well, big deal. You know, that doesn't really mean much. If this is really an active ligand, you should be able to turn on the response in a whole animal. Immunology can't be done just in cells. It's a system-wide kind of study. So if we have identified the molecular determinants for selective RIG-I recognition, we ought to be able to take this guy and turn on the interferon response at will in an animal. Now, in this regard, I should say I was completely rescued by my dear colleague, Akiko Iwasaki, who is in the immunobiology department at Yale, who saw me talk about this. And I was complaining that I was not going to work with furry animals. I was a chemist. And so she came up and she said, I'll do it for you. So the mouse work is all done in collaboration with Akiko, and now we work together on all of our projects. I do the structural biology and biochemistry, and she does the immunology and mouse studies. So Melissa Linehan and Akiko injected 25 micrograms of RNA in jet PEI. It's not just in buffer. As a tail vein injection into mice, 5 hours after post-injection, 5 hours post-injection, we used ELISA to look at interferons and various ISGs. And what we saw here was so exciting to us because you can see that these ligands that don't have all the determinants I mentioned before, single-stranded or double-stranded with no triphosphate, cause no induction in the animal. But with these little hairpins, you see a massive upregulation of interferons. And here's poly-IC, which is the gold standard for what people have typically used for turning on this kind of response. And all you need is a 10 base pair duplex. So we've proceeded from there to characterize the response a little more. I wanted to show you that the diphosphorylated hairpins are actually more active than the triphosphorylated. They're also easier to make. And so increasingly, I think that's probably going to be the workhorse ligand in further studies. And it's specific to RIG-I, and it requires MAVs. So we have various knockout animals in which to study this. So if you have a TLR7 or MDA5 knockout animal, it behaves like wild type would. RIG-I is embryonic lethal if it's constitutively knocked out in an animal. So we couldn't do that, but you can study it in MEFs. And in MEFs, you absolutely require RIG-I for the response. You can knock it down, and you can look and see that, in fact, these ligands cause specific induction that requires a wild type for these ISGs, Viperin and MX1. In the knockdowns, you can't turn that response on. So another really cool thing, so we were excited by this, and this just came out in Science Advances, because again, as I mentioned before, people hadn't been able to selectively turn on one of these kinds of receptors at a time, because nobody had actually sort of drilled down to what is the minimal ligand for one of them. So this meant that we could start to pick apart the pathway for RIG-I activation. And our first step toward that, although I got to say it's just an early step, is that we then put all of these different ligands into mice, and then we sacked the mice, we collected the organs, and we did a full transcriptomic analysis on the gene expression in all of those tissues. I'll just show you what we found in spleen. And we found the coolest things by looking at this data, and we're not really done with it. And that is that these little hairpin RNAs, SLR14 or SLR10, they turn on your type 1 interferons very, very well. They don't turn on type 3 interferons very well, interestingly, which is not clear why. However, poly-IC does a beautiful job of these. And so one of the things we're doing in the lab is we're coming up with a specific ligand for MDA5, and we're suspecting that because poly-IC can turn on MDA5, we'll get a ligand that selectively does this. And then it's really cool because some genes are turned up or down selectively by these SLRs, and you can start to understand what's really happening in the cell when you turn on this response. Okay, so to jump ahead, would these be valuable? Because the results so far suggest that if you're injected with these things, you should be more resistant to viral infection. And that was one of our inspirations. This was all being carried out as we were reading about the Ebola epidemic, and we were wondering if we could give better tools to people who are jumping in there. And so this is a type of experiment that is similar to what Melissa's doing now. There's a strain of mice that is very susceptible to flu that they use. And for example, if you pre-treat the mice with SLR10, they're completely resistant to this strain of flu. They don't lose weight, and in another strain that actually kills them, they don't die in the presence of SLR10. What's
the timing of that?
I can't get into that right now. I'm almost out. I need to use my time. But the next thing that I want to tell you about is that what we're working on now that we're very excited about is that with STING agonists and other innate immune agonists, it's been found that if you inject them directly into a tumor, what you can sometimes see is that not only do they cause reduction in the tumor by sort of inducing this inflammatory response, but when those tumor cells disperse, they have little signals that can be used to sort of vaccinate the animal against the tumor and potentially prevent metastases. So those are experiments that are going on now in the lab, and these have been done with STING agonists, and so that's something called the abscopal effect, where you treat one tumor with the agent and you look at what happens with an untreated tumor over time. So those are in the works, and we'll find out soon enough. But in the few minutes that I have left, I want to get to one really key question. I've already shown you that RIG-I can bind lots of things that don't have a triphosphate or a diphosphate. They don't signal very well, but they're in there. So why doesn't RIG-I signal all the time? How does it differentiate human RNA from viral RNA? And there are at least two parts of the RIG-I structure that aid in host recognition. One of them is a coupling motif that actually specifically can differentiate through structural mechanisms the difference between some of these host determinants and viral determinants. But the most important is the way the CTD region of RIG-I interacts with the terminus of RNA that it binds. So as it turns out, one of the most abundant RNAs in our cells are monophosphate RNAs. And so that means I have 5 minutes left. So also I have a train. So you guys may have noticed, hopefully, that in this structure I showed you before with the triphosphorylated RNAs, you can see that there are contacts to the mono and, to the alpha and the beta phosphate. So why the heck wouldn't you be able to recognize and turn on signaling with monophosphorylated RNAs? There's a lot of contacts to that alpha phosphate. So we figured it should be somewhere in between. But these are everywhere in the cell because they're the products of all these endonucleases that are chewing up RNA. So we got a big surprise when we made a whole bunch of RNAs where we just marched along and had the phosphates removed sequentially and looked at different termini. So what you see here is that in terms of RIG-I signaling and in terms of KD, and let me just explain this. This is actually, this is not a free energy. This is KD, so a big number means that it binds poorly, okay? So triphosphorylated and diphosphorylated RNAs have really tight KDs, so they're very low, binds to super high affinity. We already saw that just a hydroxyl at the end has moderate affinity, but this was a big surprise. A single phosphate is worse than nothing, right? That's just crazy because, it should be able to bind in that same pocket the alpha phosphate binds in. So we set out to understand this, and again, this part is a work in progress that's of interest to us right now. Here are two recent structures from Joe Marco Triggiano's lab where the same construct was bound to a triphosphate and to an RNA that only contains a hydroxyl. And you can see this nice network of interactions with alpha, beta, and in his case, he caught one to gamma. But what he saw is that where you don't have any triphosphates present, you don't just have a hole here in the CTD, you get insertion of a loop that's normally on one of the helicase domains. This HEL2 loop takes the place of the triphosphate. And so we did a bunch of mutational analysis of residues involved in what we think is kind of a gating interaction that prevents phosphates from binding. Oops. And just very briefly, what you see when you just do the interferon induction assay that was very satisfying is that if you take the amino acids that are in this gate and you mutate them so that now either a hydroxyl or a phosphorylated RNA can enter, you see that these mutants now signal indiscriminately. They don't signal quite as well, but they signal better than wild type on non-phosphorylated and monophosphorylated RNAs. So we think there's some gating mechanism, and because this is getting to the end of the talk, where people's attention spans are flagging the most, this is the worst time to introduce mutant cycle analysis, but nonetheless, that's what we've done, where you actually look at binding affinities where you compare two ligands with a set of mutants. And this kind of thing is helping us to pick out which amino acids are important for specifically antagonizing the monophosphate interaction. And I'll just show you that you get antagonistic, which means negative delta-delta G numbers, for amino acids that are in that HEL2I loop. So those guys are really important for discriminating against a triphosphate and selectively interacting with monos. So we think that the HEL2 loop amino acids are in some way forming a gate. We think the gate is structurally a little different in the case of a bound monophosphate, and we're trying to get the structure of that now, but time will tell. So the outlook for all of this is that we're in the process now of testing the therapeutic potential of RIG-I activators. So we're doing experiments now to look and see if they truly do have viral protection properties. Doing experiments to see if they can serve as well-defined vaccine adjuvants. This is a crying need because right now our vaccines have adjuvants in them for stimulating the innate system that are just kind of not well described at a molecular level. and also to see if these are useful as anti-tumor vaccines, particularly in combination with with checkpoint inhibitors as well. And they're great tools for doing immune pathway analysis, and that stuff is ongoing right now. So this is just sort of, to wrap up, this is just sort of an illustration of what happens if you can really see a process very carefully and pick it apart. Then you can use the information to begin designing new therapies. And I couldn't have talked about this without this wonderful team, Xiaoming and Dahai, who've done all the structural biology. Dahai is now a professor in Singapore. These guys have done the biochemical work, Dave, Andrew, and Thane. And Megan and Melissa and Akiko are our very central immunology collaborators, and I'm very grateful to them. And I'm grateful to these guys for their funding. And if you want to know more about what we're working on, you can always check out our website, which we try to keep relatively current. And I want to thank the organizers for giving me the chance to come and talk. It's a real privilege to interact with you guys, and I had a lot of fun. So thanks very much.
Thank you, Anna. So due to the limited time, we would only entertain one question from the audience. Sorry. Or two. Yeah.
Here. So what is the molecular weight of that, and is it expressed in bacteria? You crystallize it from bacteria?
I'm sorry?
Did you crystallize the structure from bacteria?
Oh, no. I mean, we synthesized the RNA on a synthesizer in my lab, and the protein is overexpressed it's recombinant from bacteria.
Okay.
But we're also making it in 293. Now we're making it in mammalian cells. We're very worried about the post-translational modifications of RIG-I and how that's affecting how it works. So we want to kind of do it all over again with eukaryotically expressed.
And did you solve the structure with that 5' monophosphate, which is very high active?
There is no structure with the 5' monophosphate. We've tried really hard and we can't get it. So we've built some new constructs so we can try to capture it. That's why we did all this mutational stuff, to figure out what was going on, because we had no other way to get at it.
Okay. Thank you.
More questions?
Thank you for this wonderful talk. Do you have any guess why would it be impossible to knock this gene out in the...
A lot of us don't think RIG-I is only an innate immune receptor. For years, we would see it in the nucleus, and now there's a paper that just came out. I never got our act together to publish it, but another lab just published last month that they're seeing it in the nucleus. You know, it's a relative of things that are involved in small RNA processing. They all evolve together. It may have dual functions. And so right now what we're designing is a conditional knockout so that we can turn it on only in the adult, because the field needs a RIG-I knockout, right? So it has to be conditional. And so that's a little more work, but now with CRISPR it's not so bad. So we need to just bite the bullet and make that mouse.
What's the role of ATP in the whole process? And can you make a mouse that has an ATP-competent binding RIG-I, but not hydrolyzable one? And what are you doing?
Yeah, there are mutations that enable a high-affinity ATP binding, but the chemistry is abrogated. That would be actually an interesting mouse to make. The thing with ATP is ATP hydrolysis is not necessary when you feed RIG-I an optimal substrate. Like those little hairpins that I showed you, they're like giving it candy. It loves those things, just like. But if you give it an RNA that's a lot longer, that can distract the RIG-I, actually ATP hydrolysis helps, and we're not sure why. I think to say that ATP hydrolysis is never required would be a mistake. We know that it's not absolutely required on the minimal substrate. But that's why a mouse like that would be really great to make.
Okay, so thank you again to our speakers, and thank you also for your attention. So now I pass the floor to Rashi for the closing ceremony.
After 4 days of excitement, science, and fun, we've come to the end of conference. So I see a lot of people are waiting for this wonderful moment of the prize distribution ceremony. So without wasting much time, I will hand over to Dilanta first, who could start the prize distribution ceremony, and then we thank our list.
So, it's on, right? Yeah. So thanks everyone for being here today for the closing ceremony, as Rashi said and already mentioned. So first up, we have the lottery. So thanks to everyone who participated and put in a ballot and visited our sponsors, and also special thanks, of course, to our sponsors who set up their booths outside and also donated a lot of lovely prizes here. So there's going to be a little bit of advertising there when I call out the prize, but yeah, it's all worth it. So without further ado, let's start with the first lottery winner, Rashi.
Katerina Combe.
Is Katerina Combe in the? All right, coming up. Not inside, so what have you won here? Let's take a look. So it's actually a thermal flask gifted to us by PicoQuant, right? By PicoQuant. Here you go. Thank you. Oh, NEB. Oh my. I'm so sorry. NEB. Yeah. Congratulations. So. Next winner.
Francesca Lugarini.
Francesca Lugarini. All right, come on up. So this is a lovely laptop bag from Sartorius. Congratulations. So let's see what Gary picks out.
I got Christian Josef Kumagum.
All right, come on up. Yeah. So you get a lovely sleeve from Sartorius. Here you go. Welcome. Front page of Sartorius' website next tomorrow.
Maria Cristina Ciccioli.
All right, coming up. That's really nice. Everyone's here. Everyone knew that they would win, I guess. So another laptop sleeve. Or you could use it for a laptop. Congratulations.
And the last lottery prize goes to
Vela Nikolova.
All right, coming up. So it's a laptop bag, this time from PicoQuant. Correctly. Says so. Yeah. Congratulations. And now we're going to move on to the winners of the poster session. So here I'd actually like to spend a little bit of time thanking everyone. So thanks firstly to all the participants who presented their research. I mean, they came from all over. Let's have a round of applause for them, please. Then I'd also like to thank, of course, our judges, from the speakers, from our faculty, and also from the organizing team. They really put in a lot of effort. I'm super happy that they took the time out of their schedules to come and really go through every poster, and I was really happy that every single poster was judged. So we'll start off actually with the results of that were decided by the judging panel. And we'll start with second place. So the second place we'll start with the first place, yeah. My bad. So in first place, we have Salma Sorabi from the Max Planck Institute for Biophysical Chemistry for her poster on transcriptional maps of the general RNA degradation machinery. So Salma, you actually have an opportunity here. You get to decide which prize you want from two prizes, not that one. So the prizes we have is, firstly, we have this Key Mission 170 camera donated to us by our sponsors, Nikon. Yeah, they hold it up. And our next, our alternative prize is a pipette set from Star Labs. And it's been signed by by our very own Nobel Prize laureates, Stefan Hell and Urban Neher. So it's been autographed. So what'll it be? What'll it be, Salma?
Guys, remember Salma is a bioinformatician. She needs the camera.
But I can never get a pipette otherwise. I'll get the pipettes.
All right, pipette set, ladies and gentlemen. So, yes. So second place, also decided by the judging panel, goes to Claudia Schmidt from the Max Planck Institute for Biophysical Chemistry for her poster titled Reconstitution of DOA-10 Mediated ER-Associated Protein Degradation with Purified Components. Congratulations. And now I'd like to thank everyone who voted using these poster ballots, because now it's time for the People's Choice Award. And this award goes to a presenter who really left a lasting impression on everyone here at the conference. So it's my pleasure to award this Coolpix W100 camera also from our sponsors Nikon to Joshua Felipe Olorosissimo from the University of Philippines, Diliman, for his poster on the psychotic effects of lactobacillus species on anxiety and memory of Danilo Rio. Congratulations, Joshua. And with that, I'd like to hand the mic back to Rashi and Gary to close this, to give you the closing address.
Thank you so much, Dilanta. Thank you all the participants, and thank you all for bearing with us these 4 days of science and excitement. I hope you had a lot of fun. You partied a lot yesterday. I know a lot of you are still drunk. Well, I am. But we survived, guys. We all survived this. So, I'm very happy to tell you a little bit about the whole year, because we started Horizons and preparing for Horizons almost a year ago. And from making a big team to dividing ourselves into small, multiple groups, we've reached here. And of course, Horizons would not be possible without these multitude of people to whom I and Gerrit, on behalf of organizing committee, would like to extend our warm gratitude. So firstly, I would like to thank our molecular biology office and Dr. Stefan Burkhardt and Kirsten Gruniger for not just giving us the opportunity, but also being with us whenever there were any kind of problems on our way. Then I would like to wholeheartedly thank George August University and Max Planck Institute for their administration support, for their financial support. As you can see, everybody is together with us, and with all full force and excitement, they made this happen. This year, we would also like to extend our thanks to MPIDS, since our new kindergarten occupied some of the rooms of MPI BPC, and that's why this year we had to shift some of the events to MPIDS, and they were very, very helpful for all the support. After that, I would like to warmly thank all our sponsors and donors. Fortunately, this list is expanding, and every year we are getting new and new sponsors, and I hope this continues for the next years. This year, special thanks to Rook and Custers for also sponsoring our breakfast. It was the first time we got a fully sponsored breakfast and multiple offers, and we are very, very thankful to all our sponsors for these multiple prizes, awards, and so on and so forth. So thanks to all of them. And
Thank you. Okay, before starting, I first have to admit that Rashi wrote most of the script for this closing ceremony and also prepared a little surprise we brought for you. So, and now, without further ado, I just want to go through all those people we haven't included yet in our acknowledgements. I want to start with Mary Osborne. Mary, many thanks for the panel discussion, for leading, and also for asking those really interesting questions which we really want to hear. And it was a nice panel. And please do not forget your MacBook charger, which is still here. Then we want to thank Peter Lösel for setting up the foyer and the poster session room, the other lecture hall. He helped us a lot for setting up and for organizing everything. And together with this, I also want to give special thanks to one person many of you have probably not seen. He's rather invisible. It is the guy in the background. It's Michael Hartig. Thank you. Many thanks, Michael, for all your support. I am quite sure without it would be impossible to run this conference. So, moreover, I also want to thank Frederick Köpper, Irene Berczakajewski, and the entire media department for your ideas, for your support, for your motivation. I mean, all of you have seen our organizers' photos, the photos they have taken throughout the conference, and also the advertisement screens they prepared together with Gabriel. So, thanks for this. Then we want to thank Uwe Krüger, the Mensa guy, for preparing all this delicious food and the coffee. So I hope you could enjoy your lunch breaks and your coffee breaks. Now I want to give special thanks to our honorary guests, so especially Katharina Seitz for providing the InDesign files for our booklet and also for giving deep and intense introduction into the program of InDesign. Special thanks again to Klaus-Peter Adam also for his support. And for for his support all the time, literally, and especially for compiling the booklet. Also, many thanks to Claudia Schmidt for the steady support throughout the entire year, and sorry for all those questions we had for you. And now, slowly going to the end, I also want to thank the support team for making photos, so especially Taras, Christina, Mark, and Jan Yun. Thank you for taking so many beautiful photos throughout the entire week. And now I have to admit that Rashi forgot 2 points in her script. So 2 persons are missing. So the first is herself, of course. So she was keeping the group of organizers together like an awesome shepherd. So hashtag Mary Osborne. Polly Metzinger, I'm sorry. So. All right, but not only this, but also for her incredible patience and her steady motivation for this, to get this 15th Horizon Week really to a remarkable event, and obviously this also was successful. Yeah, exactly. And we're thanking the organizer team as well. I give the microphone back to Rashi.
once again, thank you all the speakers for staying with us, for being there at all the times. This conference has its meaning because we had beautiful, beautiful, beautiful talks, and we just absolutely loved it to the bottom of our hearts. Then before we go on for some champagne, I would like to show a tiny video which will give you a tiny glimpse of the making of Horizons and what we were doing the whole year, basically.
All right.
Can we turn off the light?
Awesome.
Making your way in the world today takes everything you've got. Taking a break from all your worries sure would help a lot. Wouldn't you like to get away? fiance didn't show. Sometimes you wanna go where everybody knows your name, and they're always glad you came. You wanna be where you can see our troubles are all the same. You wanna be where everybody knows your name. Roll out of bed, Mr. Coffee's dead. The morning's looking bright. And your shrink ran off to Europe and didn't even write. And your husband wants to be a girl. Be glad there's one place in the world where everybody knows your name, and they're always glad you came. You wanna go where people know people are all the same. You wanna go where everybody knows your name. Where everybody knows your name, and they're always glad you came. Where everybody knows your name, and they're always glad you came. Where everybody knows your name, and they're always glad you came. Where everybody knows your name.
I forgot what I said before. 18 bottles of this red wine.
Okay.
This ET. 10 bottles of Vermentino, which is an Italian wine, which is I really like. 10 bottles of rosé. 7 bottles. This is an exact number of this other wine here. This other wine here.
Too much choice, too much choice.
All right. Which is a bit more pricey, but it's very good. So I'll just buy 7 for special people. You know? And then
Gary, do you agree with her choice?
Totally. It's really
wait, there was
delicious.
and I forgot the others. I need to look my list. I think that's
Okay.
it.
And they're always glad you came. You wanna be where you can see our troubles are all the same. You wanna be where everybody knows your name. Roll out of bed, Mr. Coffee's dead. The morning's looking bright. And your shrink ran off to Europe and didn't even write. And your husband wants to be a girl. Be glad there's one place in the world where everybody knows your name, and they're always glad you
again, thank you everybody for being with us. And now you can proceed for a glass of champagne and some juice and talk a little bit more before.